Okay, it's recording. Yay. Okay, today we're going to talk about getting ready to do PCR. So we have our primers, theoretically, and we've ordered them, and they came in. And now I talked a little bit, I think, last time that when we get primers, they come lyophilized, which essentially means freeze-dried. So we have to resuspend them. And they give us the total mass that was sent to us, so however many nanograms, micrograms, and then we always put it in 100 microliters of TE. So we'll talk about getting our primers ready. Today we're also going to talk about the template DNA that we need to do the PCR off of, which in our case would need to be genomic DNA, because we're going to try to PCR out right from the 5' UTR through the white stuff in the genome. So that's all genomic DNA. So that would be what our template's going to be. So we're going to go through as if a little bit on the protocol of genomic DNA, just so you guys get an idea of how you would do that if you had to. And then we're going to work on some calculations. I'm hoping that my iPad will connect today. Wednesday wouldn't connect, and so we just talked about calculations, which is just about the worst way to teach anything ever. So yeah, that was bad. So I'm going to have the Wednesday people watch today, and then work with them also. So let's see here. Let me share and get my iPad displayed. And I tried to set stuff up so I wouldn't spend the whole time setting things up, but best laid plans. Okay. Oh, it's already going better than Wednesday. Go over a little bit. Okay. Here's a nice blank screen for us. Okay, so let's see. What do I have first? Okay. So first we're going to look at the PCR kit and what we need. So before you want to start any kind of protocol, you want to make sure you have all of the reagents you're going to need. So we would get our GoTac Green Master Mix. So this is our, oh, hello, that's a little, I mean, I needed a fatter so I could write with this hand, but okay, let's see, try this one. Look at that, it's pretty good with no thumb. No opposable thumb on that hand. Okay, so GoTac Green Master Mix is our PCR protocol of choice because it's cheap, right? And you get lots of it, and it works really well. So that's why we chose to use this particular PCR mix. There's a million different PCR protocols out there and kits from every biotech company on the planet. And so every lab is going to probably, that you will ever be in, will probably use a different kit. But it's all essentially the same idea, and the calculations that you'll need to do are going to be the same pretty much no matter what kind of kit you use. So the first thing we want to talk about a little bit in PCR is we want to remember that PCR is DNA replication, right? And so I posted the DNA replication videos, tutorials that I had in case you only had it in 107 and nobody actually drew it out for you. I don't know how they're doing 107 now, but so that you could understand a little bit more of DNA replication and how that relates to PCR, right? Primers have to sit down, DNA polymerase comes in to extend off of those primers from that 3' end, right? So that's why we had to make sure our 3' end was a C or a G on our primer, so it's stuck nice and tight to that template, so that when DNA polymerase grabs onto it and starts polymerizing, it's going to work well. So one thing that I wanted to mention about choosing what type of PCR kit, if you're given the choice, like you walk into a lab and they say, okay, you need to do some PCR, here's the website, pick a kit. Is there's two major kinds of DNA polymerases in PCR protocols or PCR kits. And that would be I don't even know what they call regular. Well, one of them is called high fidelity. Ooh. High fidelity versus regular, and regular is almost always TAC. So TAC DNA polymerase was isolated from a thermostable bacterium. And what do I mean by thermostable? It means that I don't know if they even call it thermostable, whatever. The bacteria essentially was isolated from a ocean floor vent where it's essentially close to boiling. So it can survive in boiling water. Now that means all the enzymes in that bacteria can survive in boiling water. So boiling water, yay. I can't think of really many other organisms that can survive in boiling water. Like generally, what do you do with water if you're out camping and you have to drink out of a stream? You either filter it or you have to boil it for X number of minutes to get rid of all the bacteria and parasites and crap like that. So these specific bacteria that live in thermal vents are heat stable. They can survive. They actually like to be in that super, super hot water. And so that means all of their enzymes can function at high temperatures as well. Whereas if you think about our enzymes, our body temperature, so boiling water is 100 degrees Celsius. I actually have no idea what it is Fahrenheit. Our body temperature is 37 degrees Celsius, which is the same as 98.6 degrees Fahrenheit. And so that's how all of our enzymes essentially function. Most mammalian enzymes function around that temperature, maybe not exactly 98.6, but pretty darn close. So if we put our DNA polymerase in boiling water, it would denature and not work, right? Proteins work because of their shape. If something is denatured, that means it falls apart. It loses its tertiary, secondary structure. Still has its primary structure, right? It doesn't break those peptide bonds between the amino acids, but function is all about structure and shape. So the point I'm trying to make is that in PCR, we use heat to break hydrogen bonds. So if we have, let's go back this way. There we go. If we have double-stranded DNA, right, as is all of our genomic DNA, and you want to do DNA replication, if you're in a cell, right, if we want to do DNA replication, what has to happen first? We have to make a replication bubble. What? I totally got this wrong. Wednesday, good thing it's not recorded. What enzyme breaks hydrogen bonds and unzips the DNA?
Helicase.
Yeah, baby. So we always show it like this. Woo! Whoops. That was, well, okay, that's fine. Right? So helicase, yeah. So we could use an enzymatic way in PCR to break hydrogen bonds, but breaking hydrogen bonds by using high temperature works just so much better and so much faster. And because we're in a test tube, it doesn't matter if we heat the test tube up to 100 degrees, right? DNA is totally fine. And the only enzyme we're adding would be DNA polymerase. And so as long as that can handle boiling temperatures, we're good. So we don't need to use this enzyme. We can replace it with heat, and it essentially does the same function, but a little bit better. So in our cells, we break the hydrogen bonds or unzip the DNA so that the primers can get in there and go. With helicase, if we're doing PCR, we use heat. And so that breaks the hydrogen bonds. So we need to right, so just if we're going to copy like what happens in a cell, first we got to separate the two strands. So we do that with heat, and then the primers need to come in and they need to anneal or complementary base pair with their target sequence, just like if we had, you know, primers in our replication bubble, right? If this is our origin, right? We have our leading and lagging strands, woo, right? So they have to sit down. That's just by complementary base pairing. So the red arrows are the primers, the RNA primers that are used in cells, physiological DNA replication. And we talked about in PCR, we're going to use DNA primers because DNA polymerase doesn't know a 3' end, whether it's a deoxyribonucleotide or a ribonucleotide. And since DNA is more stable and it's cheaper to buy, we just make our primers out of DNA, right? Deoxyribonucleic acid. And they still, right, complementary base pairs, G's love C's, A's love T's, right? Doesn't matter. But we have to allow the primers to complementary base pair. So if we'll pretend like this is 95 degrees. So we actually put it at 95 degrees instead of 100. It doesn't actually have to boil to break them apart. So 95 degrees Celsius, still freaking hot, right? Breaks our hydrogen bonds. Our primers are in there. Our primers are not going to complementary base pair at 95 degrees Celsius because hydrogen bonds are broken at that, right? They have to hydrogen bond to the template in order for them to complementary base pair. So we have to lower the temperature to get the primers to sit, and that's all based on that melting temperature. So when we talked about make sure your melting temperatures are pretty close together when we design primers, that was because then your annealing or your complementary base pairing, your primer sitting down temperature will work for both primers. If the melting temperatures were super different, then we wouldn't be able to, we can only have one annealing temperature. It's both of those primers are in the tube at the same time. They both need to sit down about the same temperature. And so we're gonna say lots of primers, like 55 degrees Celsius, 58 degrees Celsius, 60 degrees, significantly lower than 95. So we have to lower the temperature for the primers to anneal. Okay, and now they're sitting down, and then the third step, now we got to get the DNA polymerase that's in there, right? He heated up, he doesn't care, he's Taq polymerase. He cooled back down, he doesn't care, he's Taq polymerase. But the ideal temperature for him to do his enzymatic work is essentially about 72 degrees if you're Taq. Ooh. So to get polymerization, I can't spell, ploy instead of polymerization, polymerization, it's about 72 degrees. None of these temperatures would work for our real cell, our human DNA replication, but all are good for Taq DNA polymerase. And so way back when, before you had a machine to do all these temperatures, people actually had little water baths that they moved their tube from water bath to water bath. So they put it at 95 degrees, and then they'd wait a couple minutes, and then they'd put it in a water bath that was 55 degrees, they'd wait a couple minutes, and then they'd put a water bath at 72 degrees, they'd wait a couple minutes, and then they'd start over. So some poor graduate student did that for, 7 hours to get through 30 cycles or something to make PCR happen. And now the machine just does it for us. We just stick the tubes in there and the temperatures go up and down. If you were using, which I'm not sure, but probably somebody tried, if you were using an enzyme that was like our enzyme, like a 37-degree enzyme, you can imagine you could have these 3 water baths, and you'd have, right, you'd have everything in there except for your polymerase. You could heat it to 95, you could cool it to 55, That'll get the primers to sit down. And then you'd have to cool it down to 37 degrees and pipette in your polymerase. Then you'd move your tube back to 95 for a couple minutes, right? You'd stick them back in 95 for a couple minutes. Then you put them into 55 for a couple minutes. Put them into 37, but you'd have to add polymerase again, because the other one died at 95 if it can only survive at 37, if you were using real DNA polymerase. And so that's super tedious, right? And even if you had a machine, every cycle you'd still have to pipette in the polymerase. So this heat stable Taq DNA polymerase is what revolutionized PCR and made it automatable. Now there's one problem with Taq. Well, there's not just one. So Taq is not high fidelity. What does that mean? It has no proofreading ability. So I don't know if they do it in 107. They're supposed to do it in 360. It can't do DNA repair. It can't check itself for mistakes. So Taq makes mistakes. Oh, I don't know what the number is, but it's maybe something like 1 in 8,000 base pairs. 1 in 5,000 to 8,000 I'm gonna make up. I think it's something like that. It's definitely not lower than that. So for every 5,000 base pairs it polymerizes, we'll say, it makes one mistake, one mismatch, right? It's something, it puts a T complementary to a G or something stupid like that, and it can't fix it. So if you're making gigantic pieces, like if we wanted to PCR something that was 8,000 base pairs long, we wouldn't want to use Taq because we know it'll likely make a mistake by probability. Now that's not to say our small piece that's 500 or 1,000 won't have a mistake in it, but probability's on our side. And it's easy to sequence through 1,000 base pairs. That's just one sequencing run, boom, not expensive, no big deal. If you want to get through 5,000 to 8,000 base pairs, that's 5 or 6 sequencing runs, which is more expensive and more of a pain in the ass. So to check for mistakes makes it more difficult. So for us, we are perfectly happy using Taq and our Go Taq Green or Green Go Taq or whatever the hell Kit was talking about, because we're not so worried about mistakes and we can sequence to make sure we didn't make any. If your clone happened to have a mistake, you have other colonies on your plate to pick to find one that doesn't have a mistake. Now, if you need proofreading because you are going to PCR, you want, you know, greater than equal to 5,000 base pairs, like when we do site-directed mutagenesis and we're making an entire plasmid through PCR, then we're going to want to use something that proofreads. So it has a proofreading ability so that if it makes mistakes, it fixes it during the PCR cycle. And so an example is polymerase called PFU. And again, it's just a polymerase that is isolated from a thermal stable bacteria that does proofreading. So we didn't do anything to make it proofread. It's just an enzyme that already does proofread, just like ours, right? Our DNA polymerase does proofreading during replication, right? It happens like if there's part of that enzymatic complex that checks for complementary base pairing. So we talked about the front end does the polymerization, and then somewhere in the middle checks to make sure there aren't any T's attached to G's. If there are, it backs up and fixes that. So that's essentially what proofreading is. And so the high fidelity polymerases will do that. They're more expensive and they're a little slower. So if you want cheap and fast, you use Taq. If you need to make sure there's no mistakes, we cannot risk a mistake in a plasmid that would totally screw up all of our experiments. We can risk a mistake in 500 base pairs because then we just pick another colony. It's not that big of a deal. And it's most likely that we'll not make mistakes. And in all our years of using this, I think out of I don't know, 50 students, one person's had a mistake. They picked another colony, it was fine. So that is your tutorial on polymerases. Ooh. So just in case you're somewhere and it's not a TAC-based polymerase and it's called something else, there's a whole bunch of different ones. PFU is just one of the ones out there. Then you'll know whether it proofreads or not. And then you won't want to choose to use TAC if you're making something that's by more than 5,000 base pairs, because it'll make mistakes and that would suck. Okay, does that make sense? Are you okay with that? No, yes, okay, I see heads. I see heads. Okay. That was long-winded, sorry. There's so much teaching to be had here, people. All right, so we got nowhere in our protocol, but we've learned. Okay, let's see. Ooh, writing on this guy. Let's see, where's, okay, that's it on that page. All right, so now I'm just showing you that. We're gonna go forward. Okay, so this is from Promega, and way back when I started using Promega, because they're distributed through Fisher, and Fisher gives us free shipping, and it's all about money, right? So science is driven by time and money, more money than time. Okay, so this is, now it's gonna be annoying. We do a 50 microliter reaction volume. And again, the reaction volumes are generally based on what type of PCR machine you have and where and what it works ideally at. And so the ones we have at CSUN are okay for 25 microliters and for 50, but we do 50 just because it's then pipetting error doesn't affect the overall volume so much, right? Like if you accidentally put in a microliter and a half instead of a microliter, it's much less change, like of the salt or something. It changes the total concentration of that mispipette in 50 much less than if it's in 25. So it's just a little safety, and the machine works great both, and these reagents are cheap, and so we're not trying to save money by putting it in a small volume when we make less mistakes. We don't do 100 because that we use lots more reagent, and that big of a volume actually affects the speed at which the whole volume of liquid heats up, because we're heating from 95, right? And then we go down to 55 or so, and then back to 72, and then back to 95. You'd wait longer to get 100 microliters up to temperature than 50. So for 50 microliters, you could set that for like 30 seconds. For 100, you need to set it for a minute. And if each cycle is about 2 minutes, then for 50, it would be about 4 minutes for 100. And so that's a lot more hours if you're running 30 cycles. So it just goes faster too. So that's how we ended up with the 50 microliter reaction volume. And so this is all the stuff we need to do PCR. So we need our master mix. We need our upstream primer, and that in our terms would be our forward, that we were calling forward, or our 5' primer. And then the downstream primer was a reverse that we made. And then the DNA template, that's the genomic DNA we were talking about that we need to make. And then we need some nuclease-free water. A nuclease is anything, remember ASE at the end is an enzyme, and usually what it does or what it chews up is part of its name. So if it's a nuclease, it chews up nucleotides or nucleic acids, and we really don't want that in our PCR, because if it eats up your primers, then you don't have any primers. It's not gonna work. If it eats up your template, you're not gonna have any template, and it's not gonna work. So we don't wanna just throw any water in there. We throw like special cleaned water to make sure it's all good. Okay, so the first thing we're gonna talk about that has anything to do with math is our master mix is set at a 2x concentration. So 1x concentration is what we always want our final concentration to be. Okay, so if there's a bunch of stuff in this master mix, which there is, which will, I think somewhere has it here, what's in there. Instead of giving us, you know, it's 2 micromolar of this and 5 micromolar of that and 4, they just say, this is the end concentration we want at 1x. What we're giving you in the tube is twice the concentration of everything. If it said 5x, it would be 5 times the concentration of what you want. If it said 10x, it'd be 10 times the concentration of what you want. And so 2x is twice as much as what we want at the very end. And so you can see if our final volume is 50, how much of 2x master mix do we want to add? Right there, it's showing you. Right, we want to add 25 microliters to have a total of 50, because that essentially cuts this concentration in half. We're diluting it in half because we want to go from twice as concentrated to just our one-time concentration. Why can't we just, why don't they just give us the 1x of that so we don't have to dilute it? Anyone? How about a hint? You have to add stuff. Here they say you're adding 0.5 to 5 microliters, depending on the concentration of, right, your primers. So let's say you added 5, you added another 5, and then in our template, we added another 5. If this were at 1x, and we wanted a 50 microliter reaction total, you'd need 50 microliters of the master mix, right? If it's already at 1x and you want 50, you're gonna have 50. Well, wait a minute, Malone. He just said we're adding 5 microliters, 5 microliters, and 5 microliters. And so now, I don't know if I can do that math in my head. It is Friday. Now, oh, our total volume is 65 microliters. Are we still at 1x? We're at less than 1x. I don't know, we're at 0.75x or something, because we added volume. You can't keep the same concentration of anything if you add volume, right? If you're in chemistry lab and they give you, you know, a salt solution, NaCl, and it's 50 micromolar in what it gave you, is it 50 micromolar if you add a bunch of liquid to it? No, it's more dilute than that. It's something you don't know. It's 45 micromolar. It's what you could do the math, but it's not the right concentration. If the right concentration is 1x, you can't add, you can't dilute it and still end up with the right concentration, right? This is gonna then be less than 1x. And we don't want that, we want 1x. So we have to start with the master mix at more concentrated so we can add a bunch of shit to it and still end up at 1x. And so that's the idea here. So if we, oh, I probably can't. I'll draw in black. So instead, we start at 2x and we put in 25 microliters, just like it tells us. And then we add our 5, 5, and 5. Now we have, oh, I dropped my, hang on. I dropped my stylus. Okay. Whew, that was tough. We have 30 and we have 40 microliters, right? If we just add that stuff. And so then are we at 1x yet? No. We have to be at 50 microliters to be 1x. So then what do? We're like, oh shit. And then that's where it says nuclease-free water to 25 microliters. Now, I'm not a big fan of the way they wrote that. I would say nuclease-free water up to a total volume of 50, right? So, because they're telling us, so I would say to a total volume, 50 total volume, oops, shit. So F, total volume, which means we're going to try to get 40 up to the 50 microliters. So how much water do we add to 40 to get to 50? Woohoo, I can
10
do that.
micro.
Yeah, right. And so that is how you get to that 50, and now you're at 1x. Woohoo! So because the company doesn't know exactly how many microliters of primer you want to add, sometimes you need to add very little of your primer. They want the primers at 10 micromolar, but in some cases you really only need a half a microliter. In some cases you might need 3 microliters. It just depends on that primer. And so they can't just say add 1 microliter of your primer, because that might not work for you. So that's why they give you this range. And same with the template. They're just telling us, oh, I can't even see it. You can believe me, under here it says less than 250 nanograms. Like, we generally make our template at least 100 nanograms to do PCR. And so they don't know what concentration our genomic DNA is at. So they can't tell us just add 3 microliters. Because if it's at 5 nanograms per microliter, that's a lot different than if our genomic DNA was at 500 nanograms of genomic DNA per microliter. So that's why these are not just, this is exactly what to do like in a regular recipe if you were making cookies, right? It wouldn't necessarily, it wouldn't tell you, you know, 1 to 5 eggs. Like, oh, how do I know, right? It tells you 2 or whatever the real thing is. Sorry. So that's why these in here are variable, because the company doesn't know exactly what all your concentrations are, because we make our own template. They don't send that with the kit. We made our own primers. They don't send that with the kit. And, you know, there could be a million different types of primers. And so these numbers I just made up as 5 each because it was easier to do the math, but it might be 2 microliters, 2 microliters, 1 microliter. That now is 35 or so, I don't know, I'm not doing the math, right? And so then you'd need 15 microliters of water. So they can't just tell us add 15 microliters of water, because it's dependent on what all the other ones are. So that's why you have to do calculations for this. And that's why we have to understand the whole 2x to 1x baloney. Or some kits come as a 10x solution, and then you have to go 10x to 1x. Okay, so if we want to do that one, let's see, yeah. Let's see, does that work? Okay, yes, hold on. Okay, so we have a 2x, oh, that's not what I want. So we have a 2x solution. And let's say it didn't tell us how much to add. Let's say I just hand it to you. And I say, I want you to give me a 1x solution, and I want 50 microliters of that. I just hand you this too. Make a 1x, make 50 microliters of a 1x solution. I remember that kind of happened to me like my first day of grad school, and I totally freaking panicked. And then I was like, ooh, M1V1 equals M2V2. So we start with 2x. We don't know how much of that we need. We know we want the end to be 1x, and we know we want our volume to be 50, because we have to be told 3 of these things in order to do M1V1 equals M2V2. There can only be 1x. Okay. And so this is our V2 is our X. So then we just solve for X, right? So we have to divide both sides, right, by 2X. And what do we end up with? Sorry, 25 microliters. The X's cancel because, right, they're the same. The 2's cancel. And so that disappears. It's X equals, and then we just divide 50 by 2 because the X is canceled, right? 1 divided by 2. I mean, so 1 times 50 is 50, divided by 2 is 25 microliters. So it's the same thing if I said it's 10x and I want a 3x solution. Now I might need a calculator. And I want 500 mLs. So we're going to do milliliters this time, right? We're making a solution. So what do we get? Somebody use calculator, do math. Because I don't want to do it.
It's 150 milliliters.
Yeah, so it's the same idea, right? X is canceled, these guys go away. 3 times 500 is 1500 divided by 10. Is that right? Ooh, did I do that in my head? I'm pretty proud of myself. All right, so you can figure out any X to any other X using M1V1 equals M2V2. Okay, so don't panic when somebody says do 10x to 1x, 50x to 1x, any of that stuff. All you just throw it in the equation. Oh my God. Okay. What else? What else was I going to tell you before we even do anything? Okay, I guess if that's good and that makes sense, so now we understand why we need 2x to 1x and what we're putting in there, just in case, right? If you just memorize what you get from one thing and then you walk into a lab and they use something that's at 5x or 10x, then you're panicking and you're like, ah! And now you're like, I got that, it's no problem. So that's why we want to understand rather than just, be a technician and do what we're told. We want to understand it. So let's go to, now we need genomic DNA. We need our template. And so they said we need, I don't remember what they said, that's right, they didn't give us volume. They said less than 250 nanograms in, of genomic DNA. So we're going to use our QIAGEN Genomic DNA Handbook. QIAGEN is I don't know, it's the best, cleanest, nicest. If you're going to make really good stuff, DNA or nucleic acids, you do QIAGEN. And it's just because I found this book. I've used Promega, it's fine. Okay. So this book actually, oh my God, sorry. This kit you can do for lots of different types of cells, as you can see here. We're going to talk about cultured cells because that's what we would use for our experiments. We would use human cell line because if we're looking for, the genes we pulled out of human tumors, so we want to use a human cell line to pull those genes out of. Now, theoretically, regardless of what human cell we use, right, that promoter sequence should be the same, right? The difference between different types of cells is when genes are turned on or off, but the actual gene is the same. The DNA is the same. It's just, do you make an RNA or not to give you your phenotype, which we talked about way back when. So we're using a human cell line that is a human embryonic kidney cell line just because it's easy to grow and it's pretty cheap to grow. And it's, so the sequence should be the same as any other type of cell. unless the problem was that there's mutations in the promoter region that caused it to be dysregulated versus something else in that cell. But we're going to pretend like that's not an option, and we look at that later. And so we're just using a cell line to isolate genomic DNA. So the kit says we can, oops, move this up a little bit. We isolate, oh, I think I deleted that page. I think the small kit says you can get 1 to 20 micrograms of genomic DNA per prep. So per tube that you're, trying to extract genomic DNA from all these cells, you can get between 1 and 20 micrograms of genomic DNA. And this gel, they're just showing us a gel electrophoresis of genomic DNA run on this gel, where we know that this would be the positive end and this would be the negative end. And we use electricity to pull stuff through essentially Jello. And then it separates by size. Since nucleic acid's DNA is negatively charged, it will move towards the positive pole. And since, it's gonna go like that. And since it's thick, it's essentially Jello, it's agar, that the big pieces get stuck in the Jello and the little pieces get through farther. So over time, the larger or longer pieces of DNA are up here and the shorter pieces are down here. So we can separate DNA by size by doing gel electrophoresis. Now these are all a bunch of smears, and that's because if you're looking at genomic DNA, that's chromosomes, right? We're talking about like little tons and tons of double-stranded DNA, millions, billions of base pairs, right? From human beings, there's 46 chromosomes. They're not packed down into what we think of as a chromosome, right? They're out in that spaghetti, right? Because we're not isolating them during mitosis, right? When they get scrunchy, they're all out in their spaghetti shape. And so as we do the extraction, we actually break up a lot of the chromosomes. So almost none of them are their full size. But you can think about chromosome 1 is huge if you think about a karyotype picture of chromosomes versus like the little Y chromosome, or chromosome 21, that's a lot smaller. So you're going to have multiple sizes in there, not just because chromosomes broke, but also because some chromosomes are gigantic and some are smaller. So you're going to get this continuum of DNA running through with the smaller pieces down here and the bigger up here. And there's really, really big and there's really, really small. It's just there's not enough of it to visualize on this gel. So they're just showing us that when you make genomic DNA, if you get this beautiful smear, that's a good thing. And you want that. And you want it to be as big of a smear as possible. All right, let's see. Oops, I gotta use my blah, blah, blah. Oh, here's where it told us. It didn't tell us yet. So here's what I was yammering about. So we would use this little mini kit, and it's between 1 and 20 micrograms of DNA that we get back. And at the very end of the protocol, where is it? We actually spin it down and precipitate it, and you get a solid. And so then we get to decide how much liquid to put that solid in. And it tells you, you go by experience and you look and see what the pellet looks like. And then you decide between 100 microliters and 500 microliters, right? Based on how big the pellet is. The bigger the pellet, the more volume you want. So where we get back to that guy, which is why they just give it to you in, they don't give you a concentration of what you're going to get back. They give you it in micrograms because the concentration depends on how much liquid you put in there. So you take your cells and you wash them, and then you lyse them, which means break them open. So that's with buffers and usually by pH. Protease is a protein proteo. A protein enzyme eats up all the proteins, right? Our chromosomes aren't just DNA, they're DNA and proteins, blah, blah, blah. Gotta get all that protein shit out of there or we won't be able to get our primers in. And then we do ion exchange chromatography. Holy shit, but we don't really have to do anything. We just stick it in this tube. And then on the filter guy, it sticks, right? All of the liquid stuff goes through, the DNA, genomic DNA is stuck on the filter. And then we wash it a couple times to get all the junk out. And then we elute, which means we add a liquid with a change of pH that makes the DNA let go of the filter and come down into the tube. And so now it's in this big volume, like 5 mils, 10 mils, which is not going to be useful for anything. And so then we want to precipitate it with an alcohol and some salt, and then you spin it, centrifuge it, get rid of all that, suck out the ethanol, let it dry, and you have solid however many micrograms, however lucky, however good you were at this. Okay, so we're going to do our first practice. And this is not M1V1 equals M2V2. I said everything we're ever going to do is going to be that, and I totally lied. We're just doing concentration. And so we're pretending like our solid was a medium-sized pellet, okay? And so we're gonna do 250 microliters of TE, and TE is just a buffer that's at pH 8, which allows DNA to resuspend better than just in plain water, which is usually under pH 7, especially if it's distilled. You get rid of all the minerals, which helps keep the neutral pH, so it's actually more acidic. So you want to use a buffer instead of just water. So tris EDTA at pH 8, and we are going to pretend that we wouldn't know this by looking at the pellet, but I'm just telling you so we can do some math. We got 15 micrograms. So my question to you guys is, what is our concentration? So how many micrograms per microliter of this guy do we have? And we're gonna do, oh shit, are we? We're gonna do something. We're gonna do breakout room and we're gonna help each other. Oh my God, what did I just do? Okay, hang on. Oh, so I'm gonna stick you guys automatically. Oh no, we don't want that many in a room. We're gonna go 2 per room, and you guys are gonna do this math. You're gonna help each other, and then I think you can just come back. And if you don't come back, then I'm just gonna make you come back. Okay, so it's just concentration. That's just what we're doing. So go nuts. Opening rooms. I think you guys can now join. Woohoo! Okay.
Why am I back here again?
Did you get kicked? Did I kick somebody out by accident?
That was ending in like.
All breakout rooms will close in 42 seconds. I swear I didn't do it. Maybe my cast did it. Okay, hang on. Oh, see? Technical issues. I don't know how to make that stop. How do I make that stop? I guess we have to try again, unless that was so fast and you guys are ready. Sorry,
Professor?
I don't know what Yeah,
Do you mind repeating the question one more time?
yeah. I'm like, I didn't mean to unshare either. I don't know what's happening. Hang on. I'm gonna blame a cast, but I think it's really me. Okay, hang on. I'm gonna say I was trying to, yeah, we're not even gonna talk about it. All right, okay, here we go. Let me see if my screen will come back. Like that wasn't supposed to go away. Come on. All right, here we go. Let's try again. Still better than Wednesday, I'm telling you. At least I think so. Maybe you guys don't think so. All right. Here we go. All these boxes, all everything. Okay, so we want to know what concentration is our genomic DNA if we got 15 micrograms solid and we added 250 microliters of TE to that. So we got 15 and we're putting it in 250 microliters. Okay, so, because if I send you to breakout, then you can't see the screen anymore, right? Okay, so I'm sending you back 15 and 250 concentration, micrograms per microliter. If you're watching this, hit pause or fast forward because I'm just going to sit here till they come back. Robbie, did you get kicked out again? Nope, she's gone. He's back.
Could I ask a question?
Yeah. By all means.
Is it a 1x TE buffer?
Yeah, it's just the TE buffer is just at its concentration because we're assuming that the solid is not taking up any volume. It will
Right.
take up a tiny bit, but you know, we're assuming that.
Okay, so I think I figured it out.
Yeah. Yeah, so we're not diluting you know, and there's no liquid in the solid. Okay, so I can't tell who's where. Because now, Is everybody? Oh, maybe I'm on the. Oh, that's because I'm right there. Okay. Sorry, I can only see Patio. I couldn't, but I'm like, but you guys aren't in your breakout room. I'm like, where did they go? And I was like, oh, now I see you. Okay. Okay, I'm gonna. Oh, is everybody back? No. All right, I'm gonna close the rooms. They have a minute to come back. Oh, and then we'll just. I do it for them if you guys okay. Because it gives them a minute. Woohoo! Everybody's coming back. Nicholas was all. Nicholas, were you all by yourself in breakout room 5? Because I think Rabia got kicked out. Okay. Everybody's back. Everybody back? Yes? All right, what do we think? What do we have to do? Anyone? No one?
Don't you just divide 15 by 250?
Yeah. See, I tried to make it seem like it's going to be hard. So if we have 15 micrograms and 250 microliters, does anybody do the calculator so I don't have to do it? What's 15?
0.06.
0.06?
Yeah.
Right, because neither of those units cancel each other, so they have to stay. And so now we're in micrograms per microliter, and that's concentration, right? Here's something good. What is this? Now, let's think back. Think. The protocol said less than 250 nanograms. So how do we convert micrograms to nanograms? Are nanograms smaller or bigger? They're
Smaller.
right? Yeah. Well, lesser, I don't know, what do you call it? Fewer, lesser, smaller? Less amount, right? So, and if we think about the nice thing about the metric system is at least all these guys are all in, the ones that we use are sets of 1,000, right? Micrograms are 1,000 bigger, I don't know how to even put it, than nanograms, right? If you go micrograms to, so then it, right? So if you go the other way, it would be milligrams, right? And since that's a bigger category, this number has to get, that number would get smaller if we put it in milligrams, and this number should get bigger if we put it in nanograms.
60.
Yeah. So what do? We go, which it turns out, we're not allowed to move decimal points in Common Core math. So I don't know if you guys missed out on that, but you're not allowed to do what I did. 1, 2, 3.
Wait, why can't we do that?
Common Core says you're not allowed. You have
Really?
to actually, you have to multiply by 1,000. You're not allowed to move the decimal.
I'm gonna ignore that.
Yeah, that's ridiculous.
Right. And that's just to make sure kids understand it versus, oh, just move a decimal. You know, 3/4 of the class just says, okay, and moves the decimal, no idea why. And then 20 years later, they're in college and they have no idea why they moved that decimal, so they don't know which way it goes. Whereas if they had to multiply by 1,000 and they remember that, they'll always get the right answer. So I get it, but it's still annoying.
My chemistry 100 professor would yell at me if I moved
If you
decimals
move the
like that.
decimal, right?
Yeah.
Yeah. Right. So, and I totally get it. And it makes sense. Like, try not to have kids memorize stuff. Try to do it by logic or reasoning, because logic and reasoning won't necessarily go away. But I don't remember formulas for math at all. Like, I remember M1V1 equals M2V2 because I do it all the time. I don't remember any, other stuff. But again, and I remember that one because it makes sense, like how it, starting, finishing. What do you want? One's going to be a variable because you know 3 other things and that sort of thing. And so, yeah, really it's like multiplying by 1,000. So if you multiply this by 1,000 or divide by 1,000, depending on which direction you're going, versus just moving the decimal 3 places. But essentially it's moving the decimal 3 places. So now let's move that guy over there. So now let's say, okay, holding this pen is, once I got it held, it's okay. Now this is so that's our concentration of genomic DNA. And let's say we're gonna put in, it said we want less than 250. So we're just gonna randomly say, let's do 200. We wanna make sure we have enough in there. So we wanna put 200 nanograms into our reaction. Oh. Right, and we have 60 nanograms per microliter. This is what we want. So how many microliters do we add to make 200 nanograms? If we want 200 nanograms, then how many microliters would we add to our reaction? And so again, this one's not really M1V1 equals M2V2 because we're not talking about molarity or concentration in that other reaction. We just need to know how much are we going to add. How many microliters? I mean, I guess you could probably do M1V1. I don't know. I have to do it on a piece of paper. All right, let's break out. And maybe it'll put you guys in different rooms, maybe. Oh, I can't find oh, there we go. Oh, we want to recreate. Let's do different ones. Okay, so then we don't have everybody in the same. So it's supposedly switch you guys up. So go to your breakout room and figure out how many microliters are we going to add to get 200 nanograms. How many microliters of this guy to give a total of 200 nanograms? Yay!
I'm alone.
Hello!
I made it.
Are you in a breakout room?
No,
Oh,
I
okay.
mean,
you and
I
you
can't.
and Leslie are together. How did that happen?
No, I mean, I can. I literally got in when you said 60 milligrams, and then you're like, how many? And I'm like, what the heck is going on?
Oh, I lied. We
That
could
makes
totally
sense 'cause I
do
worked all over
M1V1 equals
myself.
M2V2. Okay, so we want 200 nanograms in our reaction, and we have 60 nanograms per microliter. So how many microliters do we want to add?
Sure.
And you and Leslie are together, so that works.
Hi, Leslie.
Hello.
Go to your rooms.
Okay, bye.
Oh, I love saying that.
How dumb that is.
Okay, I'm going to my room now, but not
Go
because you told
to
me.
your room.
Because I want
Naughtiness.
to.
You guys are back already? Woohoo! Are you back, Alessandra?
Yep, I'm here.
Okay. I'm like, are you back or did you never leave?
No, I left pretty early actually. I left before you finished talking.
Oh, like I'm out.
Yeah. I was like, oh, whoops, I didn't mean to leave that early.
You're like, ooh, Sally. It's okay. I got my cheater math up here on top. Can you see the breakout room list?
No.
Oh, okay. I wasn't sure like what, if you guys are seeing my screen and I have the breakout room list on top of the writing, I was like, maybe they can't see it. Did you guys do M1 V1 equals M2 V2?
Yeah.
Just, okay. Because otherwise for those, sometimes you're doing M1 V1 equals M2 V2, but I'm just kind of doing it, without writing it out. Because it's, right, it ends up being just that divided by this, right? And then, oops. And then if we play cancel all your cancels, and then I don't know what that number is.
It's 0.3.
Okay, I'm like, I didn't get my calculator. And that's, wait, that should be, no. Did I fuck that up?
I think you didn't divide the what do you call it? The gram by liter.
Oh, I'm doing it backwards. Yeah. Right? Yeah, how,
Aren't you looking for X?
yeah, but wait.
Did we start off with the 60 or the 200?
Yeah. I should be doing, see, I don't even know how to do it. I only know how to do it like, I only know it's 200 divided by 60, right? So yeah. Yeah, I guess you really can't, right? So you, I was trying to make it work in M1 V1 because we want to know, this is really what we want to know. We want to know how much of this, oops, gives us, but we don't have the same 200. Oh, I guess we can, right? We just don't have that other, that one's just 1. Is that right? We don't have a V2. And so then we do this. Yeah, sorry, that works. And then you go like this. And then, and then we're left with X equals 3 point something or other.
33.
We don't do this. Right? $3.3?
Yep.
Yeah. Need more room. Let me squeeze this. Oops. Oh, it's squeezing the whole thing. There we go. All right, we're closing those guys. And then what's our, oh, sorry. I'm doing my shorthand, which I shouldn't be doing. This is microliter. And so what we're left with is microliter. Sorry, I was trying to make it work in the real M1V1, but it really doesn't because one is not, there's no final, there's no final volume that you're looking for. It's just a concentration. Okay, so is everybody here? No. Oh, we're still missing a few. So don't look at this. If you try to do it like that, then yeah, because we don't, X is not the volume of the 200 nanograms. That's not, yeah, we want how much of the other one. All right, let me move this over.
I was right. Woohoo!
Girl.
Actually, I got 3.33.
Go! Sig figs!
Yeah. I didn't see the sig figs. I didn't write anything down. I just got 60.
I have no idea what the sig figs are. I mean, it's like whatever.
It
And
is
again,
Yeah, I
true.
always
You're
fuck up
right.
sig figs.
It always depends on like how accurate your pipetter is. If it's a P20, it doesn't freaking matter. If it's a P5, then 3.33 is different than 3.3. But
This is true.
yes. Okay. Are we back? So I did it wrong the first time trying to test it out, which is the one I scritched out up here. So you can't do the standard M1V1 equals M2V2 because we're not looking for final concentration. Like we're looking for how we want to get to 200 nanograms. We don't like not 200 nanograms in a volume. We just want 200 nanograms, right? So hang on, let me make that go away. Come back here. I can't see my own script thing. And so so I wrote it out. So we have our concentration. We have 60 nanograms per microliter. How much of that, X, do we need to get to 200 nanograms? So we don't have that final volume because there isn't a volume, but it still works doing it this way. If we multiply both sides, we divide both sides by 60 nanograms per microliter, right? These guys all cancel, all of those units cancel. On this side, the nanograms cancel. We end up with microliters and we get 3.3 microliters to add. And so that's what I'm talking about. Think about logic and what makes sense. And that's what Common Core is trying to teach us too, is if you get an answer that doesn't make sense when you get to the end, like up here, I came up with 0.3 microliters, trying to do M1V1 equals M2V2, separating the concentration of the nanograms and the microliters, right? So how did I know I was wrong? 0.3 microliters of 60 nanograms per microliter. So that's less than a microliter of 60 nanograms per microliter is going to give me 200 nanograms? No, if I took 0.3 microliters of something that's 60 nanograms, I'm going to get like 20 nanograms, not 200, right? Is that, right? We're starting with a smaller, 60 is smaller than 200. We need more than 1 to get to 200, right? It's just 200 divided by 60. Right, in order to get the right amount. And then if you want to do it in a real math equation, you can do it that way. So if you're not getting, so if you think about, okay, well, if I put 3 microliters of something that's 60 nanograms per microliter, 3 times 60 is 180. That's pretty close to 200. And since it's 3.3, that would make up the rest of it. Oh my God, what's happening? Okay. So if you get to an answer at some point and it doesn't make sense, how could 0.3 microliters be 200 nanograms if your concentration, if 60 nanograms is in 1 microliter? You're told 1 microliter is 60. How could 200 be 0.3, right? But it totally makes sense. And then you check your answer by going back, doing the math backwards. 3.3 microliters of 60, 3.3 times 60 equals 200, right? So you can do the formula and then make sure it makes sense to make sure we did the right formula. So I'm so glad I made that mistake. I did it on purpose just to show you guys. No, didn't really do it. Does that make sense to you guys?
Yes.
Is anybody home? Is everybody sleeping? Oh, I got a thumbs up, yay. I can't see the rest of you because you're in the little sidebar. Oh, yay, more thumbs up, woohoo! Okay.
We got it, yay!
Yeah, sorry this does take forever, but I want you guys to really understand the math because it's not hard, it's just stuff you have to remember, and if we can use logic, then we don't forget stuff, and we don't want to panic whenever we have to do a calculation. Right? You don't have to be a math person. You don't have to feel like I'm a math brain. I'm good at math. I mean, whatever, right? I mean, if I classified myself as a kid, I was a language arts kid. I wasn't, it was just funny. I wasn't a math and science kid. Like that was not my natural, easier. And then what do I do my whole life? Math and science. But I also do a lot of writing, right? That's all language arts, so that is a good thing for me too. But yeah, so I totally get not feeling like it's just totally intuitive and the math is super easy and I don't have to think about anything or remember. Like I don't remember formulas well. I need to work things through by logic. And so I'm trying to teach that way too, so that you can feel comfortable with any kind of calculation that's thrown at you. And honestly, in a lab, you get to go look in your notes. I mean, it's not like you don't, that's why you have a good lab notebook, because you did the calculations to do your primers 3 months ago. You go look at them to help you remember, right? Unless, you know, there's a graduate student putting you on the spot, handing you a solution, here's 10x, make me 1x. Oh God, right? So, but you can still just run over and find a napkin and a Sharpie and M1V1 it, right? So, okay. It's been a long day of calculations. Okay, so here's our, let me see what else. Maybe we need to be done quickly. Okay, so this is just more protocol. I think I posted this one if you want to look up the nitty-gritty stuff. Talks about you have to have the right number of cells. You can't use too many, you can't use not enough. It's all pretty picky, just like all molecular biology is pretty picky. Talks about how to collect cells and get them all ready. All the different reagents and what they do is described in here. Which ones just resuspend, which ones are the ones that bust open the cells, which ones are then neutralizes the busting, which ones do the ion exchange, which ones are wash, all that kind of stuff. So that's not anything super, you know, we're not memorizing any of this. We're always going to get, you know, info. It all came as one big thing and I was deleting. Okay, and so then this is just another example of an agarose gel. In the kit, it tells you to take samples out so that if it goes totally wrong, you'll know where you lost your DNA, which usually we only do these analytical gels the first time you're testing a new kit, because otherwise it's a lot of work and, you know, the kits generally work. If you've got it to work before, you'll get it to work again. But it just gives you those options. Again, more analytical gels. And then they always give you a troubleshooting guide. So the first thing you do when something goes wrong is to look in the guide and see if it's one of the common things, right, that happens to people. Little or no yield, it's always this stupid genomic, it's always adding too many cells. People always think more is better, or they didn't feel like counting. This looks about right. Oops. And then, guilty. And then you get like nothing back. So you really have to count the cells and be anal retentive for this, unfortunately. More stuff, more stuff, more stuff, more stuff. The hell, how many pages is in here, people? Oh, here comes a different page. Okay. After all of that, it's 125. I'm going to show you this quick. Well, quick. I don't want to kill you guys. But this is, I don't know. I'll just show you this as an example, and then we'll start from here next time. And there's some homework posted. Don't worry about that. We'll do that together. That was posted because it was really supposed to be work on it in here, but I wanted it up there so you guys could download it or whatever. And I'll fix that. So this would be what we get when we order our primers. Okay, so whatever you named it, and then this is the sequence. Okay, here's our 3' end. Ooh, I followed the rules way back when. That was a long time ago, 2005. Okay, and remember how I said it gives us the concentration. So just like if when we did our genomic DNA, it gives you a solid. And so it says it's 0.7 milligrams. And so we convert that, we, meaning me, converted that into micrograms. And so again, we can either move the decimal, right? 3 places, or really it's multiplying by 1,000. So 270 micrograms is the same as 0.27 milligrams. So that's all that was. And then what I always have my lab students do is put this into, sorry, I just dropped my pen. Why it's giving 100 microliters of TE, pH 8, and then calculate the concentration of that, okay? And so if you do that math, you get 2.7, this is micrograms per microliter. That's just my little shorthand, right? So that makes sense. If you put 270 micrograms in 100 microliters, right? The 2 zeros cancel, and then the other 0 moves the decimal place, right? Or you just type it into your stupid calculator and it gives you 2.7. So that would be our concentration in the tube. So now the tube that was once your dried primer at the bottom, right, is now in 100 microliters of TE, right? And there's 270 total nanograms in there, excuse me, micrograms. Is that right? Yeah. 270 micrograms in there, which equals 2.7 micrograms per microliter. Do we buy that? That makes sense? Crickets? Okay. Crickets? Hello, crickets! Again, that's just putting a solid in a liquid. Gotta do the math. Okay. Then for this one, for this example, that the little scribbles that I have down here was we wanted, these were for site-directed immunogenesis. And for site-directed immunogenesis, you want 125 nanograms. Is that right? We wanted 125 nanograms in 50 microliters. And so we wanted to know how much of our 2.7 microgram per microliter do we need to add? And so that's what this is. So because this, look, I did not carry my units, which is the problem here, right? If I carried my units, this 2.7 is in micrograms, right? Per microliter. And then there's our X, because we want to know how much of that do we want to add. And then this one, this nanograms, we have to convert to micrograms, which we move the decimal place. 1, 2, 3, multiply by 1,000. And so that gave us 0.125 micrograms. So this is in micrograms because we want them to match. We want the units to match, right? We can't mix nanograms and micrograms without adding the thousands in there somewhere. So we just do it ahead of time. So this 0.125 is micrograms because we want 125 nanograms. And then 50 microliters is just the amount. That's what we want. We want that in 50. We want 125 nanograms in 50 microliters. And so that's really, I just wrote 50, but it's microliters. And so I solve for X. So X equals 2.3 microliters. So that's how much of this primer we want to put. But then we also, we don't have anything else, right? Everything else is that we just took out 2.3. Now we need to put it in a tube. And we want that tube to have a final concentration, or excuse me, a final volume of 50. So if we go 50 minus 2.3, that equals how much of the TE we need to add. So we're taking 2.3 microliters of our primer concentration and putting it in 47.7 microliters of TE. We can't put it in 50 because then our total volume ends up being 52.3. That changes our concentration. It would no longer be the 125 nanograms in that 50. Yes, no, maybe so?
Yes.
Okay. We're gonna leave it there. We'll do more of these. And we'll do a breakout room next time. I'll try to post anything that might be helpful, but otherwise we'll stop here. It's already been a long day of math. Does anybody have any questions before we go? This has been recorded. I think it's still recording, yes. So if you want to watch it again, if any of it was too fast or it was, yeah, odd, not good. And if you have any questions, you can email me. Otherwise, that's it for today. Questions?
I have a question from a problem earlier.
Okay.
You know, when you divided 60 by 200, you did something with the units because I got 33 and you got 3.3, and I feel like I missed something.
Let me see.
Because I think it was the nanograms per microliter, and so we ended up going from nanograms to microliter, from what I remember.
Okay, hang on, let me find that, and I can.
Oh, it's because it was 60 nanograms per microliter today.
Was it this one, the crazy one that I scribbled all over?
Yes.
Right. So it's 200 nanograms over 60 per microliter. I see.
Got it? Yeah. And then if you got that answer, and then you said, well, wait, that's 33 microliters of something that's already 60 to just get to 200?
Right.
That would be to get to 2000. Like, so you can sort of try, think about the numbers and see if they make sense. And then you can go back and go, oh crap, I forgot about canceling.
Right. Yeah, because I wrote it out correctly and I just didn't see that.
Yeah.
Okay.
Right, because it's the per, right. The per microliter problem. Thank you.
All right. Thank you for answering that.
Okay. All right, good job, guys.
Have a good
Thank you
weekend.
for spending your Friday doing calculations. Love you guys.
Bye.
Thank you.
Fun. Okay.
Quick question, Dr.
Oh,
Malone.
yeah.
Where are you uploading your videos for Zoom? Because I'm either blind or I'm blind.
Let me show you. Hang on. What am I doing? What am I doing? Oh, I'm trying to stop recording.